Thank you, Megan. Hello, everybody, and thank you for joining us today. As Megan said, my name is Derek, and I'll be guiding us through a discussion of key concepts in Illumina sequencing data analysis. This talk is an introduction of general concepts that can be applied to Illumina sequencing data, but it also extends beyond Illumina into the broader realm of bioinformatics. Later, we'll discuss some available Illumina tools to assist you in your data analysis goals. So let's go ahead and get started. Before we dive in, here's a brief overview of where we're going. First, we'll want to discuss experimental design and considerations that will ultimately drive our analysis approach. Then we'll jump into some key bioinformatic concepts, and we'll define the basic approaches and gain a high-level understanding of standard bioinformatics methodology, why they're important, and how they're different. And lastly, we'll discuss some available Illumina platforms and software solutions that will enable us to perform bioinformatics data analysis both locally and on the cloud. One last thing that I want to mention is that this presentation is meant to orient you to general considerations, concepts, and tools for successful Illumina Next Gen sequencing experience. You should always consult the literature and methods in your particular field to dictate your actual experiment. The scientific community sets the standard, and we just want to give you a high-level overview and context of what's available. With that in mind, let's talk about experimental design. A well-thought-out experiment can really help you downstream, and even before going into the lab and preparing samples, you should know what analysis approach and software you'll be using. So let's go ahead and consider this further. At a primary level, every Illumina sequencing experiment will consist of 3 steps, the first of which is our planning of our experiment. Then we move to our actual sequencing experiment. And finally, we conduct our data analysis. When designing an experiment, how exactly are you defining the hypothesis to be tested? This is really important because the answer will change every downstream step and drive our overall planning. It's also important to know what sequencer you'll be using or what is available to you. While each instrument provides genomic data in the form of reads, they can differ a lot in how the data is output and the amount of data that is output. For example, a NovoSeq will produce a lot more data than an iSeq, and a MiSeq can produce longer read lengths than a NextSeq. Last but certainly not least, you'll want to consider what software you'll use for your analysis. This is really important because it'll ultimately help you answer your hypothesis. When it comes to software, you'll want to know things like, is it a command line program or a GUI interface? If it's command line, you'll likely need to access a Linux server, but if it's a GUI, you'll likely need a PC with a lot of RAM and high storage. You'll also really want to consider how much data is needed and what are the input requirements for your software. And is the software expecting paired end reads or will single end reads suffice? We'll also want to think about things like what read length is appropriate, and is the software looking for any other input files? And these input files could be things like a manifest or additional ones that it requires to run the analysis. For more design and planning considerations, I invite you to view this additional helpful webinar, which gives a detailed look at planning a new sequencing project. Now let's consider how much data we would need for an experiment. Well, the answer is that it depends. Here we want to illustrate that the application and scope of an experiment drives how much data is needed. Factors like genome size, application, and power of detection all matter in determining how much data you'll need to answer your hypothesis. For example, when considering genome size, a small genome will require fewer total reads than a large genome. The type of application can also greatly affect the amount of data required, and we'll learn more about each of these later in the presentation. Lastly, power of detection can greatly change the amount of data needed, as can be the case when detecting things like germline variants, which require a lower power of detection than somatic variant calling, which requires a higher power of detection to get those somatic variants. To gain more perspective, let's consider the how much data is required question with some rough numbers and examples. Let's first think about a human resequencing experiment. In this case, we're looking at germline variants, so 10 to 30x coverage is acceptable. Let's use 30 for this example. The human genome is about 3.4 gigabases, so if we multiply that by 30x coverage, we'll need approximately 100 gigabases of data. For comparison, let's consider the exact same experiment, but this time with a different species, Arabidopsis. Since the genome is much smaller, only about 0.125 gigabases, we only need about 3.75 gigabases of data for the same experiment. Now let's consider a different application, but use the same two species as before. In this case, for a de novo assembly, we'll want to increase the coverage to somewhere around 70x. By doing so, we definitely will need more data than we did for the resequencing experiment. Of course, there's a big gap between the species, but really we want to take note of the increase of data needed by changing the application from the previous one to a de novo assembly here. Finally, let's think about RNA-seq or gene expression experiments. Let's again consider humans as our species. Since our goal is to analyze transcript expression, the genome size is not really taken into account, and the coverage will depend on the sensitivity you are looking to obtain. However, we can say we'll need somewhere between 5 to 100 million reads per sample for this example. You've heard me broadly mention coverage. I'm not going to talk about how to decide what coverage is right for your experiment, but I will link you to our coverage calculator in the resource section of this presentation. Before we discuss types of reads and read lengths, let's go ahead and define a read. A read is the data that comes from a single cluster on the flow cell. And this is the actual genomic sequence found for a particular sample. It would look something like this, where this read is approximately 50 bases long. This means that if it would take 50 cycles from the sequencer to produce a piece of information regarding this sample. In this illustration, we're showing that as a cluster lights up, the sequencer is able to determine what base is present at a given cycle. During FASTQ generation, we are able to put all the cycle information together to form reads. Reads are stored in FASTQ files, and demultiplexing refers to assigning specific reads to their correct samples based on their index or index combinations. I will discuss the structure of FASTQ files later in this presentation, since they are used as input to many analysis workflows. Now that we've defined a read, let's talk about single versus paired end reads. In the following examples, I arbitrarily selected 100 base pairs as the length. Single reads probe the insert in one direction from 5' to 3'. You may recall that during library preparation, we will flank the insert with adapter sequences on both ends. The insert is the portion of DNA that comes from the organism of interest and should not include the adapter sequence. I'm saying should because if the insert is smaller than 100, you may actually sequence adapter. However, if the insert is longer, then you'll only sequence up to 100 and miss sequencing the remainder of the insert. In that case, a longer read length like 125 or 150 may be beneficial. Now let's think about paired end reads. When using paired end reads, we will sequence in the 5' to 3' direction, and then after turnaround chemistry, we'll also sequence in the 3' to 5' direction. We are essentially reading through the middle of the insert. We typically sequence symmetrically, meaning read 1 and read 2 will be the same length, and again, in this case, 100 bases. There are a few exceptions, but normally they are the same. And here, this last scenario, we can have paired end with overlap. For this example, we change the read length to 200. The insert could be around 300 bases, and with a read length of 200, there would be a 100 base pair overlap region between bases 100 and 200. You may want to do this in an effort to cover the region further. However, you may not want to do this if your analysis pipeline can't handle that overlap. Deciding what kind of reads will depend on the pipeline that you plan to use and its requirements. Now that we've discussed the types of reads, let's talk about a few applications for the different types. For SNP detection or simple resequencing, we can use either paired end or single end. The key factor here really is the depth of coverage. And one intrinsic advantage of paired end reads is that you get twice as much data if the reads do not overlap. However, this may not be required for your experiment. So if you get enough from single 100 base pair reads, let's say, then it is not necessary to use paired end. Switching over to insertion and deletion detection or general structural variant detection, paired end reads work best. As we move away from the point features like in SNP detection, the structural features get much bigger. Paired end is the better choice to detect these big changes because these features in this category can be 2, 20, or even 200 nucleotides in size. So having a read pair can really help us determine how the sequence is arranged. Similarly, we'll also want paired end reads for de novo assembly. In this application, we're essentially putting together a big puzzle and determining how all the reads go together to make a genome. So having information like how close or far read 1 and read 2 are from one another can be really useful in figuring out a novel genome sequence. Moving on to RNA Seq or expression analysis, we can use either paired end or single reads. Older algorithms may not support paired end reads. However, if you're looking to detect fusions, paired end reads are required. Paired end reads can also be useful when looking at novel transcripts and investigating exon-exon boundaries. Once again, here is an example of where the software selection in the application is really important in our planning. Lastly, for small RNA differential expression applications, we really only need single end reads. The main reason is that small RNA is typically only 30 to 40 nucleotides in length, and it is so small that you wouldn't need paired end. Reading fully in the forward and then fully in the reverse would not give you any new information, so single reads are really all you need. We've talked a little about read length, but let's consider this further. The main takeaway here is that different features in different applications work best with certain read lengths. We've already discussed these applications, so it's no surprise that smaller read lengths work best for small RNA and RNA expression. In the middle range, from approximately 50 to 100 base pairs in length, we're seeing RNA and resequencing applications are each appropriate. Anecdotally, within the 75 to 125 base pair range, we get a good amount of data that can be very convenient for alignment to the human reference. At 150 base pairs or greater, we can start detecting indels and structural variants quite nicely. We can do de novo assembly as well, and probably go even a little longer than 150 base pairs, since most de novo assembly algorithms tend to work better with longer reads. The longer length is also useful for structural analysis of genes and gene annotation because you can look at larger chunks of DNA and determine how things are rearranged or particularly structured. Again, these figures are general guidelines meant to acquaint you with when to use certain read lengths. Your scientific journals or papers are your main resource for a particular experiment, and we refer you to those when doing your planning. With all the experimental design considerations behind us, let's get to the main portion of this talk, which is a discussion of key concepts in bioinformatic analysis. Of course, there are several flavors of analysis that can be performed. However, here we're looking to discuss basic concepts like alignment, variant calling, assembly, and RNA-seq analysis. At this point, we've planned our experiment, sequenced, and now we have our reads. So we're ready to move on to our analysis step. In this section, we'll first discuss FASTQ files and how they're structured. We'll then move on to our resequencing applications, focusing on whole genome sequencing and targeted sequencing. We'll follow that with our assembly applications, and we'll wrap this section up with information about RNA sequencing. So the first file and input to pretty much all analysis pipelines is the FASTQ file. And the FASTQ file for a sample will contain all of the read sequence for that sample and look something like what you see here on the screen. Each read contains 4 lines of information, and here we're looking at 3 reads. The first thing to notice is the header on the first line of each read entry shown. The @ symbol tells us what instrument was used, and in this case, it's a HiSeq. We also get additional information like run ID, flow cell ID, lane, tile, our XY coordinates, and any filter information for the read. The last entry in the header line is the index sequence as shown here, and this is a single index read. If it were a dual index read, it would look like this. The next line is the actual genomic sequence read that was read on the instrument, and this is the most important piece of information, and will be the sequence that gets aligned, assembled, or whatever the downstream analysis plans are. The next line is the delimiter. The character used is a plus sign in this case. The last line shown here is the actual quality score shown in an encoded format. Each character that matches up with a corresponding base call has an associated ASCII number, and then we subtract 33 from that number to obtain the Phred score for the base call. This example only shows 3 reads. However, an actual FASTQ file will contain millions, possibly even billions of reads, depending on your design considerations. If you take the number of reads and multiply by read length, we'll obtain the number of base pairs. For this example, we have 3 reads times a length of 66, which means we have 198 base pairs of data. Now that we understand what's in our FASTQ file, let's talk about resequencing applications. Resequencing applications refer to alignment or mapping the reads to a reference genome. After alignment, we then perform variant calling or detecting where the samples differ from the reference genome. When we say resequencing, we typically refer to whole genome or whole exome sequencing. Targeted resequencing, like in amplicon or enrichment workflows, refers to performing alignment and variant calling on a very specified region or regions of the genome. And this gives a rough outline of the general steps in any resequencing workflow, where we have our FASTQ files as input. Those are aligned in our alignment step. BAM files are output, which those are then used for variant calling. And then we finally get an output with our variant call file and our genome variant call file, which we'll discuss these a little bit more here. So let's first start by talking about alignment, which is the first step in the resequencing application. As mentioned on the last slide, alignment refers to taking sample reads and mapping them back to the reference genome. Once alignment completes, you'll have BAM files as output, and the .bam extension designates the sample as a binary alignment and mapping file. Some considerations to take into account are that alignment can be computationally expensive because of the number of reads to align. And in one of our early examples, we calculated that some experiments may produce millions or even billions of reads, and that's a lot of reads to map. So a good aligner must be able to handle this scale of data. Additionally, aligners need to make decisions on reads that align to more than one region. Reads will also have variation due to DNA features or multiple variants in a sample. So this can be tricky for aligners to handle since the goal is to map back to a region. So the better the match, the more likely a read will get assigned. Finding where the samples are different is also important, and the aligner needs to be able to map to the correct region even when there's high variability. So to understand the alignment task a little further, let's consider this simple example. Here, let's say our reference genome is the sentence, aligners might place a short sequence in many places in the genome sequence. If we have short reads like in and n, they may get us aligned here, but they can also get aligned here. If we increase the read length and get the reads in the and enum, we can see that these reads only align to one spot right here. If we increase the length, that may not always be the best answer. And if we have place and sequence, we can see that those reads should align here, but they can also align here. In that last case, paired end reads may be more appropriate. So here we have the read in ... place, and that can only align in one spot here. Lastly, for this example, aligners need to determine what to do with this variation. So in this example, we have the read placed. And as we can see in that sentence, it's nowhere in the reference. However, the read may fit here if there's a deletion of the D. And it can also align here if we have a SNP that results in a change from S in the reference to D in the sample. Now that we have an understanding of alignment, let's briefly talk about targeted alignment. Just as a quick reminder, we use targeted alignment in experiments where we only want to consider certain regions of the genome, like in an amplicon analysis. Let's say you have a reference genome, but your experiment is designed in a way that you're only interested in these two regions. The leftmost region may look like this, where it is only about 30 bases long. And the rightmost reference region may look like this with a length of about 50. In this example experiment, we don't care about other areas of the genome. We're only interested in these two. We already mentioned that reads could align to more than one region in the reference. So how can we ensure that our aligned reads only fall in our targeted regions of interest? The answer is that we can use a manifest file to filter our alignment results to show us only the reads that fall within our target regions. I don't want to get too out into the weeds with the manifest file, but what I do want to provide is a quick example of the key information in the file that helps us with targeted alignment. We'll need to indicate a reference genome as seen here. And here we're using HG19. We'll also want position information like chromosome and the start and stop coordinates. And lastly, we'll need reference sequence information to match up our targets. After we complete our alignment and use our manifest to determine our reads on target, we can visualize our data like this. In this visual example, we can see our paired end reads where our forward read or read 1 is shown in red and our reverse read or read 2 is shown in purple or blue. The read pair is aligning to one of our target regions. We actually see several pairs aligning to the target region in this example. And now that we have all this set up, we're ready to go ahead and discuss the next step in resequencing applications, which is variant calling. We've already alluded to variant calling a bit in the previous examples, but let's go ahead and dig into this a bit deeper. After completing alignment, we want to understand where the samples are different from the reference sequence. We can have small point differences like in SNPs or larger deletions and insertions of full sequences. We want to make sure we are detecting these variations as much as possible in our samples. Again, as mentioned earlier, the output files are VCF or variant calling files and GVCF for genome VCF files. The difference in the two is that the VCF will only contain information on variants identified, and the GVCF will include information for all positions, including where the sample matches the reference, also known as a ref call. Considerations for variant calling include depth or coverage, meaning how many times have you sequenced the region where the variant is found. As you would expect, the higher the coverage, the more certain we are of the variant. We'll also want to know if we're looking at low frequency variants like cancer variants, somatic variants, or germline variants. Once we have our variants, we'll want to annotate or understand information about them, and then interpret or make determinations about what the variant means based on the annotation and your specific experiment. So let's think about coverage for a moment. As we said before, greater coverage increases the confidence we have that the sample sequence is correct. In this example, we have our reference in gray and our sample reads in blue. We located a variation in this position here, and we have a C instead of a reference A. Since the depth of coverage is 1, we can't be really sure if this is an actual variant. It could very well be a SNP, but it could also be a sequencing error or even a read that was not aligned to the correct region. To be certain, we really need more data or reads to come to any kind of conclusion. Now we have this scenario. Again, we have the reference in gray and our reads in blue. However, now we have a coverage depth of 4, and we are seeing that C being called many more times. We now can be more sure that C is the correct call and that a variant is actually present. This would be appropriate for a germline or SNP case, but let's go ahead and take a look at a somatic case. Again, we have the same setup as before, but instead we have just one read showing the C, while the other reads are displaying an A or ref call. If you're using a germline variant caller, this C may be disregarded and it would not be called a SNP. However, if your study is looking at low frequency variants, like in a cancer tissue biopsy, this one read with a C call may be very important as a data point. Again, this depends on your study design and understanding the software you're using, and in this case, the variant caller being used. Let's go ahead and switch gears now and talk about de novo assembly. So for de novo assembly, we're looking to build a reference genome or understand how the genome of an organism may be arranged. Our output files will be FASTA files, and they may contain a range of contigs. With assembly, it's really important to understand that you may not get one continuous genome in the first try. Assembly is an iterative process, and it could take several attempts and parameter adjusting before obtaining a good reference. Considerations will include the approximate size of the genome you're working with, the continuity of the contigs obtained and the organism. And by organism consideration, we mean, are you expecting for the genome to have a single chromosome, like many bacterial species, or will it have multiple chromosomes? Of course, an organism with several chromosomes will result in more contigs. However, it is also possible for a single chromosome organism to have several contigs as well. During library preparation of de novo assembly experiments, we expect to fragment the DNA in such a way that we'll obtain a variety of insert sizes. The mix of long and short inserts will help us better cover the genome. The longer inserts will help create a backbone, while the shorter ones allow us to sequence the middle regions of the longer inserts that we missed. As we'd expect, the reads for each sample will contain, or the FASTQ files for each sample will contain reads from all regions of the genome. This makes it even more important to have paired end reads or a complementary read 2 for each read 1 to best assist with our assembly task. The FASTQ files will then run through the assembly program, which align the reads in a sequential manner. We'll then get a FASTA file which contains the assembled genome or contigs to better understand how the reads go together to form a genome. I do want to clarify that this is a very simplistic model just for example purposes. To get a genome assembled into a continuous FASTA, it's really important that you consider the literature available and abide by the specific parameters used to get the best results. And as previously mentioned, those may have to be revised in order to produce the best results. Now, let's think about a completely different experiment and talk about RNA Seq. There are many types of questions that can be answered with an RNA Seq experiment, but most commonly we are looking to measure RNA expression and abundance. Here's a high-level diagram of the process. We obtain our sample, extract our RNA, convert it to cDNA, and construct a library and sequence. Once sequencing is complete, we obtain FASTQ files. In this most common case, we are looking to find gene expression. This equates to how abundant a particular gene is as opposed to another for a particular sample. The sample could be in the same sample at different time points after a treatment. And in that case, we would take a plant at time 0, add a chemical or the treatment, and measure what genes are expressed half an hour, or even 2 hours after the treatment. Basically, in this case, we are counting what genes are expressed or not expressed over time for that plant sample after we've added our chemical or other treatment. Like in resequencing experiments, RNA Seq begins with alignment. However, alignment is done differently. Let's say we're looking at a transcript, and in this case, the gray line here is the reference, but the boxes represent the exons that we're interested in. During alignment, we will want to align only to these exons, so the intronic regions will need to be removed. So single reads can align to a single exon like this, and from that, we know that this first exon is present, and so is the second one with 2 reads aligning. You can also have single reads that span 2 exons. This branching can indicate a deletion of the intronic region. With paired end reads, we get more information, like an insert where read 1 aligns to this exon and read 2 aligns to this other exon. Also, we can see an instance where read 2 spans an exon-exon boundary. Ultimately, we'll get information about how many hits you get on an exon, and we'll also learn about exon-exon boundaries or exon associations from paired end reads. Lastly, once your alignment is in place, regardless of whether single or paired end reads were used, we'll need to count these hits to answer our questions. So let's talk about how to do that. There are a couple ways that we can count expression, but here I'm only going to show the simplest. Let's say that we have this scenario. The treated sample has a depth of 5 for each exon, and the control has a depth of 2, but that's only part of the story. Let's say for the treated sample we get 50 million reads, and for the control we have 10 million. We need to normalize them if we're going to compare them. We can normalize by millions of reads. In this case, for the treated sample, we divide our depth by millions of reads and get 0.1. If we do the same for our control, we get 0.2. So even though at first glance we see more coverage and reads in the treated group, when we normalize, we actually observe half the expression levels for treated versus the control. Normalization is very powerful, and most RNA-seq software will do this automatically. In this case, we would do this between samples. Now let's go ahead and look at another case. In our previous case, we were looking at ways to compare between sample and sample. Now we want to compare transcripts from within the same sample. So here we have 3 different genes, each of different sizes, 2, 1, and 3 kilobases. Let's go ahead and throw in our aligned reads, and we get a raw read count of 4, 5, and 9. But like the previous example, that's not enough. We need to go ahead and normalize. So in this case, let's standardize by read length. So 4 divided by 2 is 2, 5 divided by 1 is 5, and 9 divided by 3 is 3. Now we have normalized by transcript length within one sample. Lastly, let's talk about how these 2 methods are combined and used for normalization. Reads per kilobase of exon per million of mapped reads, commonly known as RPKM, takes into account reads mapped and length of the transcript for normalizing single reads. Fragments per kilobase of exon per million of mapped reads, or FPKM, normalizes in the same manner as RPKM, but is specific to paired end reads. Since RPKM and FPKM are normalized in the same way, these counts can be used to compare samples with one another. Transcripts per million of mapped reads, or TPM, is a different method that can accommodate either single or paired end reads. Overall, RNA-seq fragment counts can be used as a measure of relative abundance of transcripts. And for more information about RNA-seq analysis, we have an entire webinar covering that topic. And I will link that out to you in the resources at the end of the presentation. That concludes our introduction to key concepts in bioinformatics. Like I said earlier, there are a lot of other analysis methods that we did not cover, but our goal was just to get a very basic picture of what's available. Now let's go ahead and look at some Illumina tools and solutions that will help us in the analysis process. First, I'd like to briefly talk about Local Run Manager, more commonly known as LRM. LRM is a service that runs in the background of the Windows operating system. It is present on board the iSeq, MiniSeq, MySeq, and the NextSeq 500, 550 instruments. LRM can also be used off instrument when installed on a PC that meets all the computing requirements and can be used to analyze data from any of the benchtop instruments that I just mentioned. LRM uses modules to perform data analysis. Here we are only showing a few, but more can be found on our LRM support site. There are modules for all types of applications, including resequencing, targeted resequencing, assembly, and several others. Modules do need to be downloaded and installed separately as you need them. Here are a few links to get you started with LRM, including installation and module guides. If you'd like more information, we'll also have an we do have an LRM recorded webinar that can be viewed at any time. BaseSpace Sequencing Hub is our cloud-based solution. And to access BaseSpace, you'll need to create a basic account at basespace.illumina.com. Once you have that account active and you're on the main dashboard page, you'll be able to navigate to different sections, including our apps, to view all of our app offerings. Once in the apps page, you'll see a variety of Illumina, BaseSpace Labs, and third-party apps. Illumina apps have an icon with a blue background. BaseSpace Lab apps will also have the same blue background, but will feature a flask in the upper right corner. And third-party apps will have unique icons in a variety of colors. On BaseSpace, you'll find many Illumina apps that are very similar to the workflows and modules that we have in Local Run Manager. And they perform a variety of the same tasks like the ones we talked about earlier. Here are a few links to get you started. And like in LRM, we also have a recorded webinar that guides you through using BaseSpace and all of its features. I will link you to both webinars in the resources section of this presentation. Okay, and next I would like to briefly discuss our DRAGEN platform. And broadly, here are the various analysis pipeline that DRAGEN offers, including the ability to process base call files or BCLs into FASTQ files at a greatly accelerated rate compared to conventional CPU-based demultiplexing at FASTQ generation. And our DRAGEN platform offers very accurate data, which is able to detect small variants with high analytical sensitivity and specificity. In addition to our FASTQ generation, we recently have implemented lossless genomic compressions, where we can reduce FASTQ sizes up to 5 times with our DRAGEN Aura lossless compression or original read archive. And one really important thing about our DRAGEN platform is its robust speed, and that it can process an entire genome, human genome at 30x coverage in about 25 minutes. I'd also like to highlight that DRAGEN has frequent releases that include regular enhancements and upgrades. And this image just gives us a very basic outline of everything that's available. But there are many applications, and with each release, new applications and pipelines are available. In addition, we have recently implemented many tools that support the SARS-CoV-2 data toolkit. And our DRAGEN pipelines are available on on-premise servers, but BaseSpace as well, and then additionally on our NextSeq 1000, 2000 platform. So with that, our NextSeq 1000, 2000 platform integrates the DRAGEN platform on board and offers many more tailored analysis pipelines, including those seen here, the DRAGEN Amplicon, COVID-seq test, enrichment, FASTQ generation, RNA pipeline, our single RNA pipeline, GATK, germline, and then we have additional ones that can be run in BaseSpace if you don't want to run them on the NextSeq 1000, 2000. Okay, and we'll go ahead and move on to our summary and conclusion. So today, we covered our design and planning considerations. And what I want to highlight is that we want to focus on the amount of data that we need, whether our experiment will need single reads or paired end reads, and the read length that we'll need. Again, there are several considerations. I invite you to look back at those slides. Those are three of the most important. We also talked about analysis concepts very broadly. Specifically, we talked about resequencing and alignment and variant calling, and then de novo assembly and RNA Seq applications. Lastly, I gave you a quick overview of some of our solutions for data analysis. Again, there are several more, and I invite you to visit our support site to see anything else that might enhance your experiments. But we talked about Local Run Manager, BaseSpace Sequence Hub, our DRAGEN platform, and our NextSeq 1000-2000 with onboard DRAGEN. As promised, the slide deck does include a number of resources, like our coverage calculator, method and kit selector, and several tech notes, as well as general information about FASTQ files and demultiplexing resources. I mentioned several additional recorded webinars, and we have some information here to help you access any of those that you may be interested in. And we also, on our support site, do have a number of upcoming webinars listed and information about registering for those webinars. For any additional assistance, please contact us at the link below. And I do want to just note a couple of upcoming webinars that you may be interested in. Our amplicon sequencing and assessing the success of your run. So this would be a very good follow-up to our discussion today. And then also our iSeq 100 webinar, which will discuss run quality and best practices for that instrument. And that brings us to the end of our presentation. Thank you very much for your attention, and I would like to open the floor to any questions in the chat.